So I'm an internist, but I have a subspecialty training in infectious disease, and I've worked in hospitals and also worked for Bristol Myers Squibb doing drug development for quite a few years. So I'm going to share with you some of the perspectives on drug development in nosocomial infections. So you say, nosocomial infections, what is that? Hospital acquired. So you got that infection into the hospital. So the next slide I'm actually going to show you. All right, so nosocomial infections, they're a global problem. There's not one country that's not affected by hospital-acquired infections. And hundreds of millions of people get these infections every year. Now, there's been a relative decline in the US because over the past 2 decades, we've put in projects to focus on reducing hospital-acquired infections. And one of the biggest things that we've done is wash your hands. You can't go into a hospital without seeing hand sanitizers, and you see signs up that say wash your hands. This is the most important thing that's helped decrease the number of hospital-acquired infections we see in developing countries like the US or the UK or in Europe. Now, even though those have been relatively successful, 7 out of every 100 hospitalizations, somebody's going to get a nosocomial infection. In fact, a couple of years ago, the CDC did an evaluation of hospital-acquired infections. 1 in every 25 hospitals, somebody's going to get an infection today. Now, outside in the future developing countries, China, India, Brazil, the incidence is even higher. So 10 out of 100 hospitalizations, 10 out of 100 people that go in the hospital are going to get an infection. And not only is it associated with people dying and bad things happening, like prolonging your hospitalization or having to put you on antibiotics, it is extremely costly. The CDC estimated a couple of years ago that it is about $36 to $45 billion a year between prolonged hospitals, all of the infections that can lead to other things happening to you. And that's one of the issues that we're trying to make better. Now, the next slide shows you, well, where are these infections coming from? I said hospital acquired, but anytime you go into an institution, rehab, just had my knee done, going into rehab, it's a possibility you could be exposed to bacteria there and get an infection. Clinical centers, outpatient centers, so any of those could be areas that you're being predisposed to getting an infection. Well, how does it happen? We spread it. Doctors, hospital staff, visitors coming in to see patients all have the potential of exposing you to bacteria. And why is that happening? The widespread use of antibiotics. I got a cold, not feeling well, going to see my doctor. Probably is viral, but you may end up with an antibiotic. Go home with your antibiotic. And most of the times we don't know what might be causing it, so I don't give you an antibiotic that's very narrow and only kills a few bacteria. I'm going to give you an antibiotic like a cephalosporin that kills a variety of different bacteria. And empiric treatment. Well, empiric just means I'm going to treat you before I actually know what's causing it. And that leads to widespread use and very broad spectrum antibiotics being used. Well, who's at risk? Well, the group that's most at risk is if your immune system is any way compromised. Now, the immune system means those white blood cells that are floating around in your body, they happen to see a bacteria come in and they kill it. That's what they're there for. But if you get chemotherapy or something else that decreases the number of white blood cells that you might have, it puts you at a little bit higher risk that if you get an infection or if you're exposed to the bacteria, it might cause an infection. As we get older, or the very young. Our immune systems are not functioning as well as they did when we were my daughter's age. Post-surgery. So anytime you do a break in the skin, you open up an area where the bacteria can get in. That wasn't meant to be a rhyme, but it actually does happen that way. IV catheters. So I go in the hospital, an IV might get placed in, I did a break in the skin. If there's bacteria on my skin, try to clean it with alcohol and all of that, but the likelihood is there's potentially a risk associated with that. Last week I was at home and I went out for my walk and I saw my next door neighbor. And she was walking around and she was on a cane and she said, you know, I had my knee replaced about a month ago. I said, well, I haven't seen you in a while. She said, well, I ended up in rehab and then I got discharged and I went home and I was looking at my scar, the knee, and it was starting to get a little tender and a little bit red, and she ended up having an infection at the site. And it was because she was diligent about it and went in to see her doctor that it was able to be controlled. So same thing happened to you. It can happen. And that's one of the things I'm going to talk about. So what can we do about it? Before I talk about what we can do about it, the next slide just shows you there has been a change in how bacteria and the relationship they have with antibiotics. So for the past 70 years, antibiotics have been used very effectively to treat infections. It's reduced people dying from infections, it's reduced some of the morbidity associated or the bad effects after you get an infection. And when, for example, when penicillin first came into the marketplace, bacteria were very sensitive to it. You know, you got a strep throat or an upper respiratory tract infection due to strep pneumoniae, gave you penicillin, and it was very effective in improving the infection. Today, what has happened is that because of the widespread use of antibiotics, and remember that empiric therapy I talked about, where we're using broad spectrum antibiotics, it's increased the resistance bacteria have to the antibiotics, some of the antibiotics we're using. Well, bacteria are very smart. You know, I expose them to a little bit of antibiotics, and the next thing you know, they develop mechanisms to resist being killed by that particular antibiotic. And in fact, there was a survey, although it's hard to really get a handle on what's the incidence of infections due to these resistant bacteria, the CDC estimated that in 2013, over 2 million people in the US had an infection due to a resistant bacteria. And that caused about 23,000 deaths. And as a result of that, preventive targets have been put in place between Health and Human Services, public health, hospitals. They've been very aggressively, at least in the US, working very aggressively to try to reduce the incidence of nosocomial infection, especially due to these drug-resistant bacteria. The next slide just shows you how does this resistance spread? Well, in the center is the antibiotics. Now, antibiotics can be given to animals that are our foodstuff. Sometimes in very low levels of antibiotics to prevent the animals from getting sick or to treat an infection in the animals. You know, cows, chickens, all of that good stuff. Now, when the animal's exposed to the antibiotic, gets in their gut, the bacteria in their gut are exposed, and they can develop resistance. Animal goes to slaughter, we kill it, some of that bacteria might get on that meat. And if we don't cook it properly or wash properly our hands as we're preparing it, it has the potential to get in our food as we eat it, and the next thing we know, we're colonized. It's in our guts. How many of you have heard of E. coli being on spinach or lettuce?
Oh, yes.
Oh, yes.
Strawberries too.
Strawberries too. You know how it got there?
Not washing hands.
Well, the fertilizer that was used on that lettuce or those strawberries might have been made from the feces from these animals that had resistant bacteria or any kind of bacteria. That's how they get on our fruits and vegetables. The other way that bacteria can get into the environment, the community, let's say George or Mary, whomever. George is the guy that I was just saying wasn't feeling very well, went to see his doctor, had a cough, a little bit of fever. He got an antibiotic. Now maybe George wasn't diligent and he didn't take the full course of the antibiotics. How many of you have taken the full course of antibiotics when you're given it? How many of you don't? Sometimes we don't. I mean, after about 3 or 4 days, it's like, oh Lord, I don't think I can take any more of this, and we stop. Well, what happens when you do that is that if you did have that bacterial infection, we didn't treat it long enough to get rid of the bacteria, and the bacteria, being very smart, gets a little bit of exposure, it develops resistance mechanisms. And the primary resistant mechanism that they often develop are what's called enzymes. They produce these enzymes that break down the antibiotic, so it's not effective. Now, George might have 2 things happen to him. His infection could get worse, or let's say George slips and falls and breaks a leg and ends up in the hospital. Now, he's colonized with these bacteria. He goes into rehab or he goes into the hospital. People are going in and interacting with George, and the next thing they know, on their hands, they picked up his bacteria, and it may spread it to other patients. Or George is fine, doesn't go into the hospital. He just goes in and stays in the community. Now, George isn't very good when he uses the restroom. Oh, come on. And he touches something.
Tell the truth.
Tell the truth. You need to know, because that's the way you can start to protect yourself. Wash your hands. Use a paper towel when you touch that handle. Now, the next slide shows you how this issue is creating laughter in my family, but also how the CDC is really focusing on these resistant bacteria. In all seriousness, they have 18 different bacteria that have been categorized based upon how much resistance these bacteria have and how much they're causing a problem. So the urgent threats are the bacteria that have resistance and are causing a bit more problems in terms of infections. How many of you have heard of antibiotic-induced colitis? Okay. Clostridium difficile is the is the organism that's causing this. And it often happens after somebody's been on a broad spectrum antibiotic for a while. This develops resistance and it could cause what's called a colitis. We've also seen some forms of Neisseria gonorrhea that are becoming very resistant to penicillins. Now, the serious threats are those that are we still have antibiotics to treat them, but they are causing problems. How many of you have heard of methicillin-resistant Staph aureus?
Sure.
Right? Sure, everybody's heard of it. And especially in New York, quite a few years ago, it was big in the news. And we do have an antibiotic to treat those called vancomycin. It's held in reserve, which means you can't use it unless you get an approval, usually by an infectious disease person that has to come and evaluate the patient and say, yes, you can use this particular antibiotic. The other one, which was probably in the early mid-1990s, drug-resistant TB was making a resurface in New York. So the typical antibiotics that we were using to treat TB, rifampin, streptomycin, those sorts of things, the bacteria were becoming resistant to. Fortunately, we've seen a decrease in that over the past few years, but that had the potential to become more of an urgent threat. It's been better controlled. And then those that are concerning threats are the bacteria that really haven't caused a lot of infection yet, but are very worrisome because we're running out of antibiotics to treat them. One of which is vancomycin-resistant Staph aureus. So remember that methicillin-resistant Staph aureus I just talked about? And the fact that we still have vancomycin to treat it? few select one of strains of them are becoming resistant to vancomycin. So we need to be aware of this only from the standpoint of what do we need to do drug development against? Which are the bacteria that are really becoming problematic, and how do we get a handle on that? So let me talk a little bit about what treatments are in development, but before I tell you what, the next slide, what treatments are available, let me tell you about drug development to begin with. Now, first of all, developing a drug, from the time you identify this is a potential drug, this goes for any drug, not just antibiotics, and begin to develop it in terms of understanding how it works in the body. How does it get distributed? Are there any toxic effects? Is it effective in the disease that you were developing the drug for? It's about 10 to 15 years. And not only is it long, but there are large numbers of drugs that are start out in the laboratory that will never make it to get reviewed by the FDA for approval. They either aren't as effective as we thought they were going to be compared to what's currently available. They have some toxic effects on the body, and they just don't make it through to the stage of which you've got enough clinical data, enough safety data, enough efficacy data to have it reviewed by the Food and Drug Association to say, you know, we believe that this drug is something that needs to be marketed. So by the time you go through the long development cycle, the clinical trials, the drugs that fall out of bed, it's about $1.4 billion to develop and bring a drug to market. This was estimated by the Tufts Center for Drug Development, and they released that study in 2014. So it's very costly to do it. Now for antibiotics, the next slide. From antibiotics, I'm going to talk specifically about drug development for antibiotics. Now there's two paths you can take in developing a new drug, new antibiotic for the treatment of an infection. The first is, on the top, it's novel. There's no existing class of antibiotics currently approved. In this case, there's a lot we don't know about that particular drug. First of all, we have no idea, because it's totally new, what the side effect profile, what the potential toxicity of the drug is. And we try to make sure, first, to do no harm. So you don't want to try to develop a drug that's going to potentially have a really bad effect on the person you're giving it to. We don't know, in the case of an antibiotic where there's no history, is resistance going to develop quickly with this particular antibiotic and therefore make it useless? Is it going to be efficacious? And in what types of infections can it be used for? And what organisms will it be active against? So the hurdles, if you go down that route of developing a totally new novel antibiotic, the hurdles are pretty high for that one to actually get developed and get enough clinical information to where it could be considered for approval. Now the other route, which is the one that's been done most often, is that you have a class of antibiotics, let's say penicillins, to do something novel with that penicillin. You change its profile a little bit so it's able to kill that bacteria in a different way. And so that bottom level, the hurdles are a little less because you know pretty much what the toxicity of the class of antibiotic is. You know a little bit more about how it might attack the bacteria, and maybe it has some additional benefits in attacking that bacteria. But because it's a class of antibiotics that has been used, the potential that resistance might develop more quickly because bacteria have seen some form of this antibiotic might be higher. So in some ways we reduce the hurdle, but in others it may be a problem. At the end of the day, getting it from the lab, which I call the bench, to the patient, which is at bedside, it's going to take a while and it's going to be costly. So the next slide, just shows you what I meant by the drug development being a little challenging. So in the 1980s, this is courtesy of the FDA, the 1980s, there were a lot more new drugs coming into the marketplace. So from 1980 to 1984, there were about 20 new antibiotic applications that the FDA approved for new antibiotics. Fast forward to that last little bar, 2010 to 2012, there were about 2. Which means that the number of new antibiotics being developed and approved for treating infections, including drug-resistant infections, it's giving us fewer and fewer options. Now the next slide. Well, why is this such a problem for antibiotics? I talked about all drugs having long development cycles and it's very costly. Well, there's some antibiotic-specific challenges. First of all, the molecules or the drugs that we're developing, a lot of them can be broken down. They're against the enzyme targets on the bacteria. Remember I said that the bacteria are very smart. They develop enzymes to break down the antibiotic. Well, this can also mean that resistance will develop quickly. So I might get it approved and I might show it's effective, but when that drug has been out there for 3, 4, 5 years, what's the potential that resistance is going to happen? There's also an issue where these novel antibiotics, there's some commonality that bacteria cells and human cells might have in common as far as the target. So that drug might also affect you or me. And that's where the toxicity comes in. So that's an issue. And then once it's approved, it's sometimes held in reserve because if it's going to be effective against these resistant bacteria, there may be a need to put it on the shelf until I really have to use it. Now the next slide just shows you also some of the additional challenges that happened in the late 1990s to the early 2000s. There were 3 new antibiotics that were approved. And unfortunately what happened once they got on the market, there were some side effects that required them to be pulled back. Now that occurrence, in conjunction with concerns about widespread use to begin with, caused the FDA and the FDA equivalent in Europe, called the European Medicines Agency, to change their guidelines for drug companies to develop antibiotics. The guidance got a little harder to meet. The clinical trials required for you to do got bigger, longer, more costly, made it a little more difficult to enroll patients, and also there were issues with once that drug got approved, as I've told you before, it was held on reserve. So this made some of the bigger pharma companies decide to put their resources somewhere else instead of developing more antibiotics. And that was one of the reasons, if you go to the next slide, that we're seeing more of these companies moving away from developing antibiotics. They're developing more drugs for cancer, for neuropsychiatric diseases, for all of those, but we're seeing a little bit of paucity in the numbers that are being developed for infections. So a lot of the companies now that are doing this are biotech companies. They're smaller, they're more nimble, they don't have as much overhead. And so about 80% now of new drugs being developed that are antibiotics are being done by biotechs, which is actually not a bad thing. They're also getting very creative with technology. So they're using what's called bioinformatics. How can I use technology to better understand the genes and how these bacteria reproduce so that I can find ways of treating it better? They also have better mechanisms now to screen the drugs that are being considered for development. So if you go to the next slide. So there's still hope. Well, one of the things that's happening, there are more and more partnerships that are occurring between the biotech companies and the larger pharmaceutical companies, which helps. The focus on technology is good, and at the end of the day, this whole focus on using technology to understand gene sequences allows them to get a little more targeted in the type of antibiotics that are being developed. If you go to the next slide. So in the past 6 years, remember when I showed you in 2012, there were about 2? Well, in the past 6 years, there have been about 8 new drugs. They're all for serious infections, so all the nosocomial, hospital-acquired infections. They also have to be given by vein, so again, for serious infections. Some of them are skin infections, some are for intra-abdominal infections, upper respiratory tract infections, and some for the Clostridium difficile. There was one for Clostridium difficile that was becoming more resistant. Last year, there was an evaluation of how many new antibiotics are in development. There are a total of 48 being developed now. That's the good side. The challenge with that is about 16 of those are in very early stages, so they haven't even gone into clinical testing yet. So we don't know how many of those 16 are actually going to make it through that development cycle. There are 15 out of the 48 are what's called late stage, which means they are in clinical trials being evaluated for a variety of different infections. And hopefully, some of those will make it through. They're also being developed in classes of antibiotics that we already know about: penicillins, erythromycins, and cephalosporins. The next slide. So prevention is still the biggest way we can prevent these infections. Education, awareness, and a lot of collaboration between healthcare workers, between hospitals, clinicians, quality improvement organizations, and the government, like the CDC and HSS. The next slide really focuses on, all right, so we talked about the health environment, we talked about hospitals and all of that, but an ounce of prevention is worth a pound of cure, literally in this case. So what can you do? Wash your hands is probably the biggest thing. You see the signs all over, you see the hand sanitizer, but it's really true. It is really true because you can pick up on the surface anything you touch. Prevent the spread of foodborne illnesses. This is one that I really like to emphasize, because if you practice good food prep and how we do it, that also can decrease a lot of the infections or the bacteria that we might get colonized, might get in our system. First of all, wash your hands, but also the surfaces that you're using to prep your food. I'm gonna make a salad. The bag says triple washed. I don't have to wash that lettuce. True or false? False. False. No, not true, false. Because once you put the lettuce in that plastic cellophane container, you've just created the perfect moist environment for the little bit of bacteria that might have still stayed on after they finished doing that triple washing to grow. I don't need, true or false? I have an orange, I'm gonna cut it, and I don't have to wash that before I cut it. Yes, you do. Because in that skin could be the bacteria. You cut through it, and now you've introduced it into your orange that you're just about to take a nice juicy taste. I am preparing my food, and I'm just cutting through that chicken. My cell phone rings. I wash my hands before I start the process. I go grab my cell phone. Problem or not? Problem. Problem. But I've seen that happen where people are preparing and you forget that I need to wash my hands again before I go and pick up my cell phone. Don't cross-contaminate. That's an easy one to fall trap to. You're cutting, you're hurrying, you're preparing your chicken. Next I'm gonna go cut up that broccoli. It wasn't my intention. Or I cut up that chicken on a cutting board and I didn't really clean that cutting board all that well, and now I take my broccoli and I'm cutting it on the cutting board. You just cross-contaminated without really even knowing you did it. Cook your food to an internal temperature of 160 to 165 degrees for most meats, fish, and other. Yes, that, blood red steak might be good, but if you don't cook it well, if in the event that meat has something on it you don't want on it, cooking it at least helps decrease the potential. So I've cooked my food, I've done my safe prep, I've cooked my food, and now I'm going to leave it on the stove for a little while. Problem or no? Problem. Because as soon as you take it out of that oven and it starts to cool down, there may still be some bacteria on it. Probably kill them all off, but you let it sit out there for a little bit. What's going to happen? They start multiplying again. So chill your food. So it's not just the mayonnaise. Everybody's heard about the mayonnaise and somebody had the potato salad at the picnic and they left it out. So it's not just eggs and mayonnaise. It's also meat and other things, that chicken, that roast chicken that you had. Who likes stuffing? I mean, yeah. So these are all things where you cooked it in an area where probably it's going to get exposed to some bacteria. So these are simple things for which we all can take action against, keeping these bacteria from taking a foothold, colonizing us, or even exposing ourselves in the event something like a hospitalization or that knee replacement surgery and you go home, whatever. Those are things that we can do something about. Questions? Ben?
So I'm encouraged to know that the biotechs are taking over some of this, developing antibiotics from the pharmaceutical companies. Do you see a day where maybe they'll be able to outpace the bacteria's ability to mutate and resist every antibiotic that's out there?
So it's a good question. I think that development is a part of it in terms of getting new drugs and it's trying to stay ahead of the curve, or at least to address the bacteria that we know are threats now. But prevention is probably the biggest way to reduce the incidence, and prudent, what's called prudent stewardship, which means really restricting the antibiotic to a documented bacterial infection, being more prudent on how widespread an antibiotic. Do I need to use a hammer for that nail? Or can I use something a little bit smaller? And I think there was a comfort level in trying to go very broad spectrum because you knew you'd kill whatever it might be. We got to get more careful about how we use antibiotics. So I think outpacing, it's a combination thing. It's doing more drug development, which the biotechs are really trying to get a handle on along with pharma, but also trying to be more prudent once the antibiotic is developed. Any other questions?
I also heard that your kitchen counter sponge is the worst culprit.
It is the worst culprit.
And people sometimes you can put it in a microwave for 10 seconds to kill it. If you do that every day, then that might help cut down.
And it's even better if you can use the more sanitary wipes,
Yeah.
so that you can really, because they're also going to be able to have something in them that's going to kill the surface. It is those sponges, because you think you're doing a great job by cleaning off the surface and putting a little bit of whatever dish detergent or whatever on it. It doesn't do enough. And those bacteria, because the sponge is wet, they like moist environment, it's sitting at room temperature, they just grow. They just grow. So it's really thinking about those sorts of things in our homes where we can in the community, because a lot of these some of these resistant bacteria you're reading them or you're hearing about occurring in the community. That person didn't even go into the hospital. So, any other questions? No? All right. Everything you need to know about drug development.
Thank you.